Everybody for coming. So we have this public lecture. This is the first lecture of our Society for Neuroendocrinology. It's nice to see lots of guests here as well, who are here to hear wonderful Anna Marie talk about her research. I'm going to hand over first to Susan, who's going to
So hi everybody, warm welcome to me as well. It's my real pleasure to do this It's a brief biography of where I'm from. So Anna received her degree in biology from Southampton, and she received a PhD from Southampton as well. And Anna was interested in human disease, the biology of human disease, for the honest background. And she worked during her PhD on gut and working on molecular biology of T-cell lymphoma. She moved on as a postdoc to Salisbury, where she investigated molecular biology of T-cell exposure. She's been in Exeter since 2005, where she is currently a professor of human genetics, and she's also director of research and impact. Now, Anna is an expert and a renowned work leader in the biology of reproductive aging, and we are really going to talk about the biological plot exploring the genetics of humans.
Thank you, Sujin, and thanks to the organisers for inviting me to talk today and sharing some of the work that we've been doing in our team here in Exeter, looking at the genetics, particularly of human reproductive ageing. So there are two really key events that kind of mark the reproductive lifespan in women, and that's puberty and menopause, and between those two time periods is what we often think of as the reproductive lifespan. But actually, we know that across the lifespan, there are things going on that are determining a lot of these events, and they're not the only key milestones. So I'm going to talk a little bit about the whole of reproductive aging, which happens across the lifespan in humans. Now, humans are actually quite unusual among the animal kingdom in that they spend a large proportion of their life not being able to reproduce. And when you think about it, this is kind of counterintuitive to how we're supposed to work. We're supposed to be here to reproduce. And so to spend a third or up to a half of your lifespan not being able to reproduce doesn't seem to be like a very sensible thing to do. And in the animal kingdom, there are only a handful of other species that spend as long a proportion of their lifespan not reproducing. And I've shown a few examples on the slide. I don't think you'll guess which ones unless you already know. But of these three other marine mammals, only killer whales and beluga whales have a long post-reproductive lifespan, whereas the dolphin, which is part of the same family as the killer whale, does not. So the hypothesis is that actually that menopause has evolved as a positive selection event, and a lot of work's been done actually here in Exeter looking at killer whales and the sort of understanding that process, and it seems to have a positive selective advantage. So that's an interesting sort of sideline to the story. Now back to humans, no more talking about whales, I'm afraid. So back to the human story. And we know that the age at which women go through menopause varies quite substantially within the population. So this graph just shows you the distribution of menopause timing in a population, an average population. This is actually data that's come from a study called the UK Biobank, and this is from over 60,000 women. And you can see that the average age is around 50 years, but there's a huge spread. There are people down here going through menopause in their 20s, right up to in their mid-60s, still haven't been through menopause. So there's a lot of variation. And one of the things that we're interested in our group is the impact of this timing on health. We know that women that go through menopause later are at increased risk of some of these cancers, particularly breast, ovarian, endometrial, colorectal cancer. And we think the reason for this is because if you go through menopause later, you're actually exposed to estrogen for much longer in your lifespan. And estrogen is a hormone that can predispose you to these cancers. So that doesn't seem great. But if you'll go through menopause earlier, there are also health risks. So certain other cancers. Probably the most prevalent health risk, though, is osteoporosis. So here again, we think estrogen's important. And so going through menopause earlier means that women will have less estrogen over their lifespan, and estrogen's really important for good bone health. So there are health implications for earlier and later menopause. And so one of the things that we do in our group is to try and understand that relationship much better. I'm actually not going to talk too much about that any further, but just to let you know what the relationship is and one of the drivers for the research that we're doing. So the end of reproductive lifespan, the timing of menopause, is very much governed by what we call the ovarian reserve. And this is the number of eggs or oocytes that are present in a woman's ovary. Now, humans are actually born with all of the eggs that they will have for their lifespan. They're laid down during fetal life. So this diagram here shows you a timeline from conception, here on the left, through birth, puberty, up to menopause. And against that timeline, plotted the number of eggs that are present in the ovary. So you can see that the maximum number of eggs that a woman produces, about 7 million, are actually made before she's born, about midpoint of gestation. Then what happens is you get this massive waves of atresia or cell death, and the oocytes get destroyed in the ovary. So that when a female baby's born, she will only have about 1 million eggs. These then decline further up to the point of puberty. And it's at puberty that eggs start to be ovulated, released from the ovary every month. But in fact, that release of eggs doesn't actually account for a very large proportion of that 7 million that we start off with, because in a woman's lifespan, she'll only actually ovulate 400 or 500 eggs. So the large majority of the loss in eggs over lifespan happens by this atresia process, which is a picture of it's at the bottom here. And when the number of eggs gets down to a critical level, and we think that's about 1,000 eggs in the ovary, that's when a woman will go through menopause. So it's all very much tied to this, the ovarian reserve. And one of the reasons that that's really important is because of the link between the ovarian reserve and fertility. So on the left here is that same data that I just showed you in the previous slide, plotted slightly differently. So this might be, for one individual woman, the number of eggs that she might have prior to birth, and then that atresia decline up to the average menopause age here, about 51. And plotted onto here, what would happen to her fertility at those various ages. So the optimum fertility is around in their 20s, for a woman in her 20s, but then it decreases from about 30, certainly from 35 onwards more dramatically, such that by the age of around 40, so about 10 years before a woman goes through menopause, she will stop being naturally fertile. So that's a really key point. So this becomes really important as women are delaying having their families. So on the right, this is a plot showing you data from the Office of National Statistics, showing data from 2013, which was the first time that it was recorded that there were more women having babies in their early 30s than in their 20s for the first time in history. So there's definitely evidence that women are delaying childbearing. And it's also associated with an increase in the use of in vitro fertilization techniques in order to achieve a pregnancy. So that's definitely a reason. It's often linked, obviously female infertility would be linked in many cases to the ovarian reserve. So as well as looking at that reproductive lifespan and the end of reproductive life, one of the things that we're really interested in is the symptoms associated with the menopause transition. And this is important because around at least 70% of women will experience symptoms around the menopause, and many of them will find them very debilitating. Probably for 10%, these symptoms will go on for 10 years or more. Women often report having to give up work. And obviously this is a time in the early 50s when many women are at the peak of their careers. They may give up work. It can affect relationships, and marriages break down, and so on. And this is some data from a survey from the police service, actually, from 2019, which is of over 6,000 women, asking them about what symptoms they report when going through the menopause. And I think most people are aware of hot flushes. So 72% of women reported hot flushes, but there's lots of other things associated as well. Joint and muscle aches, frequency of needing to go to the toilet, irritability. So lots of things that have been linked to that menopause transition, which can affect quality of life and ability to work. So that's another area that we're interested in investigating and to use genetics to understand. So the reason that we know that genetics is important for these conditions, things like menopause timing, menopausal symptoms, we've known about this for many years, in fact, way before we knew what the genes were. And we know about it from studies of families and particularly twins. So when you study twins, you can look at, you can compare identical twins and non-identical twins. And if for a particular condition, trait, disease, identical twins are more similar to each other than non-identical twins, this is an indication that there's a genetic component to that particular condition. So we know that the timing of menopause, for instance, is probably about 50% down to your genes. And we've known that for a very long time. It's just understanding what those genes are that we haven't been able to do until recently, and I'll tell you the results of some of our research on this in a moment. So genes come in different varieties, and those different varieties can have different effects. So if you think of a gene as a fruit, so here we have an apple, you might have the Golden Delicious, and you might have a Granny Smith variety. So they're both apples, but they're slightly different. And so the Golden Delicious variety of your gene might actually cause you to have a slightly earlier age at menopause, whereas the Granny Smith might make it a little bit later. And we know that it's not just one gene that's involved. We know that there are many genes. So some women might have a whole bunch of menopause lowering fruits/genes. Other women might have a whole bunch of menopause increasing genes, and others will have a mixture. And really, it's like getting a deck of cards. You're dealt a hand during your conception and the production of eggs in your parents, eggs and sperm in your parents, to determine which of these sets you might get. And the set that you get will influence where you are in this distribution of menopause age. Not completely, of course, because remember I said that genetics only actually accounts for about 50% of this variability. But it will contribute. What about the non-genetic things? Well, we know about some of them, but not others. So this timing of menopause is what we call a complex trait. So this is a condition where we know there are genetics involved, but also other non-genetic factors. We think being a smoker has a big effect on menopause timing. So women who smoke generally go through menopause 1 to 2 years earlier than those who don't. We have fairly good evidence for drinking being important and timing of puberty. There are also lots of studies out there that have reported lots of other risk factors. I think that those are more questionable. The evidence is still slightly weaker, but things like exercise, weight, and other environmental factors. So we've got a lot to understand, but we certainly know that they are contributing to that timing of menopause. So if we put that together with the genetics, what we see is that that set of risk factors will determine where a woman falls on that distribution. So a woman that has a full hand of menopause lowering genes, is a smoker, has a high alcohol consumption and went through puberty early, might have her menopause at 40. Whereas a woman who has a hand of menopause increasing genes, is a non-smoker, doesn't really drink and had a late puberty, she might go through menopause at around 60. So that's the factors that we're thinking about, but we only know about a proportion of them. We don't know the identity of all of them just yet. So I'm going to focus for the rest of the lecture on the genetic contribution to these traits, particularly to menopause timing. And this is a diagram of the human genome. So we have 23 pairs of chromosomes, and this is them lined up in a sort of diagrammatic form. And then women will have 2 X chromosomes. And the DNA, the chromosomes are made of DNA, and we have about 3 billion bases or nucleotides or letters in the DNA code, if you like, in the human genome. And this code codes for around 20,000 genes. And those genes are spread across the chromosomes. And what has happened in recent years, in the last 15 years, is that the technology has moved on so dramatically that we've actually been able to start to uncover some of the genes that are involved in these complex traits. We've known for many years about some of the single gene disorders, things like cystic fibrosis, Fragile X syndrome that I worked on previously. We've known about those for many years because they have a single gene cause. But these conditions that are complex and have multiple genes plus the non-genetic factors have been much harder to get a handle on. And what we had to do in the past was to make a guess about which gene might be involved. So we might pick a gene that codes for a protein that's made in the ovary, and we test it in some people perhaps who had very early menopause and see whether they had any alterations in that gene. The problem with this is that it's a very slow approach for a start, doing one gene at a time. But probably more importantly, it really hasn't found very much. And that's true for a lot of the diseases and conditions that we study in human genomics, that this candidate gene approach has been pretty unsuccessful. So everything changed for this complex trait genetics field in 2007, when a new technology became available, and this is called genome-wide association studies, or GWAS. And what this did was to, rather than sequence individual genes, it looked at markers across the genome, or flags. So we would look at different spots across the whole genome. So we could capture the variation that's happening across the whole genome, and we can do this in a single experiment. And this allowed us to look at things in a hypothesis-free way. We didn't have to make any assumptions about what genes might be involved in this condition or any conditions. We could take all of the genome in one go. We've also now moved on from GWAS and been able to explore more at the sequence level, the individual bases. And the reason that that's become important is because although GWAS was very powerful, and I'll show you some results in a minute, it only really codes for things, it captures things that are relatively common. So those variants, those 2 different types of apple, if one of them is present in 1% of the population or higher, we were able to capture it with the GWAS. But for things that were rarer, it was more difficult to capture. And these rarer things actually turn out to be really important because they often have a larger effect on the thing that you're trying to measure. So the problem with sequencing, we've been able to sequence genetics for many years, but it's always been really expensive. So when we started sequencing at the genome level, we picked the region of the genome that we thought would be most appropriate or most likely to be associated with diseases. And that part of the genome is called the exome, and this represents only about 1 to 2% of the whole genome, and this is the region of the genome that actually codes for the proteins. So that's where we started. And the costs for that were relatively cheap, still a lot more than doing a GWAS, but cheap enough that we were able to do this at scale and find new genes. But what's happened very recently, in the last, really year or 2, is that the costs of sequencing the whole genome have plummeted. And so now it's not actually that much more expensive than doing the exome to do the whole genome. And so that's where we're going, and that's where the future will be. And with the beauty of this, of course, is that we can capture all of the different types of variants, the common ones and the rare ones, across the whole genome in one experiment. And this is going to be really, change the way we see genomics and its links to disease. So I'm going to move on now and talk about some of the findings that we've made and start with the discoveries from GWAS. So we talked about the different variations that you can have in a GWAS. Well, the main one that we use for this type of study is the most common variation that's present in the human genome. And this is a single base change or a single nucleotide polymorphism. Some people call them SNPs. That's a kind of shorthand for it. There are 10 million of them in the human genome. So this is where a single letter in the DNA code is different between individuals. So you might have a C at this position in your genome, whereas the person sat next to you might have a T. For most of these variations, it doesn't actually impact you, your health or anything that of your features, but they are little markers of the genome. Because there are so many of them, we use them as markers. And in a GWAS, what we do is we can test for those 10 million SNPs in lots of people, hundreds of thousands of people if possible. The more, the better. And what you see in a population, some people will have two A's, some people will have two G's, and some people will have a G and an A. And then you can look at the condition that you're trying to find a genetic link with, in this case menopause timing, and see what's the average menopause age for the GG, people with GG, the average for the GAs, and the average for the AAs. And if it's different between the different genotypes, these are genotypes, then that's a suggestion that this particular SNP is flagging this region of the genome as a region of the genome that causes a change in menopause age. So we've done that with all of the SNPs in the genome, and we did that in collaboration with many other groups across the world, but it was led by us in Exeter, by Dr. Kate Ruth, who's in the audience, and I was a postdoc at the time, now a lecturer, and we collaborated with a group in Cambridge on this project and led this study. We were really excited because it got published in Nature in 2021, and we even had the front cover of Nature, which was especially exciting. And these are ovaries, not hot dogs, I would hasten to add. But still, we were pleased. Terrible picture, but we were very pleased to have the front cover. They did the design, by the way, not us. And what we found from this study was that there were 290 signals, those flags across the genome, that were linked to timing of menopause. And I've plotted them here. This is what we call a Manhattan plot. So each of the dots on here is one of those SNPs. And they're laid out across this axis here, depending on their position in the genome. So these are the different chromosomes here. So this is a whole genome horizontally, if you like. And then the position vertically is determined by how strongly they're associated with menopause timing. So we put a threshold on here, a line here, and anything that passes that threshold indicates a position of the genome that's linked to menopause timing. So there are 290 of those. And this was really exciting to find so many signals. And we found that each of those signals actually only changed menopause timing by generally a few weeks, with a few slightly over a year. But what we're seeing is, as we see with lots of these complex traits, there's lots of genes, most of them having relatively small effect on menopause. And this is why they've been really hard to find in the past, and we need these very large sample sizes to find them. So even though we've got 290 of them, it still only explains 10 to 12% of the variation in that variation across the menopause distribution. So we know there are more things to find. But that keeps us in work, so that's great. So yeah, but we're hoping to expand our studies and find more of those signals. So I'm going to move on and talk a little bit more about what we find from the exome sequencing now. So this is where we'll actually sequence the individual bases, but in the exome. So it's called the exome because we're sequencing exons of a gene. So a gene's made up of exons and introns, and the exons are the bits that code for protein. And you have multiple exons which are joined together to form the template to make a protein. And what we have focused on are two different types of variation that can happen in this exon. The first type is and the reason actually I should go back a step, that we are looking at this exon only is because we can really predict what impact some of these genetic variants are having on the protein. So the two types we've looked at are the type that actually cause no protein to be produced, and these are called loss of function. Or there's a second type where we can change an amino acid in the protein, and that could potentially alter the function of that protein. It could be effectively loss of function, but it could actually have a different function, and these are called missense variants. So we look at both of those types. And because these are relatively rare types of variation, because they have such a big effect on the protein, we group those together. So we group together all the loss of functions and test those in one group, and we can test the missense as well. And we look at the effect of those on menopause timing in relatively large populations. So we've done our first study of this, and again we did this in collaboration with the group in Cambridge. And these are the findings from that study. And unlike, the GWAS where we had 290, we only found 9 genes from this experiment. But we've only looked at the UK biobanks. We only have 100,000 women, which is half the number we had in the GWAS. And as I said, these are much rarer types of genetic variation, so they're much harder to find. So I'll just talk you through this plot. As I said, there were 2 types of variation. We have the loss of function, and they're shown in red here. The missense, the amino acid changing ones, they're in blue. And then we've also looked at what happens if you combine both together. And we're looking for deviation from this 0 line. So if that group of variants has no effect on menopause, the dot and the bar will be crossing this 0 line. So you can see we have 3 different genes where particularly loss of function variants actually are to the right of that line. That means they increase timing of menopause. And this one's particularly interesting, this CHEK2 gene. So loss of function variants in this gene increase menopause on average by about 2.5 years. And this is actually a gene that we did pick up in our GWAS findings with the common variants, but in the GWAS, the effect size was much smaller. And that's because we were looking only at a flag of the region, not the gene itself. So this allows us to get much more evidence that, first of all, this is the causative gene, and to understand the mechanism. It seems here loss of that gene, so loss of that protein is really the key thing that's driving the increase in menopause timing. The other one I wanted to just point out from this figure is this one down here, the ZNF518A gene. Now, this was a completely new gene. It's never been linked to anything to do with the ovary before. This was very exciting because look at the effect size here. So this is actually reducing menopause age, carriers with loss of function, reducing it by about 6 years. So this is actually the biggest single gene effect that we've been able to see reported in the literature. It's not actually, these loss of function variants are not carried by very many women. So this number here tells you how many people are carrying it. So in that over 100,000 women from the biobank, only 28 carry that loss of function variant. So this is what I mean about them being rarer, but they have a much bigger effect on the timing of menopause. So by putting the 2 types of study together, we can really get an idea of what the causative gene is, what the mechanism is, and really understand some new biology that's linked to ovarian aging. As I said, we're just about to move into the whole genome sequencing era, and that's very much for the future. And the reason that that's going to be really important is because we'll be able to look at not just the coding part of the genome, but also all the rest of the genome, the other 98%, that is involved in regulating those genes. Because obviously every cell in your body carries the same set of genes, but what's switching them on and off is also determined by some of your genetic sequence. So this is just showing you a diagram of how DNA in the cell is highly coiled, and in order to make a protein has to be uncoiled, and just a small part of it codes for your RNA and then proteins. But there are all these regions associated with that process, promoters, enhancers, which are DNA sequence dependent, which really govern that process and do the gene switching on and off. And we think that's going to be a really rich source of understanding some of the biology associated with reproductive aging. So how does knowing about the genetics help us with knowing about reproductive aging? Where do the findings come? And there are three areas that I think are really important and that really we've been informed about as a result of these sorts of studies. Firstly, we can uncover new biology. So, for instance, that ZNF518 gene, that's a gene that's not much is known about it, never linked to ovarian biology before. And that really could be a whole area of research, understanding what that gene's doing, how it's impacting the ovary. We can use genetics to highlight potential targets for treatments. And in the case of ovarian biology, this could be perhaps targets for infertility treatments. And I'll show you an example in a minute, some of the work we're doing to look at menopausal symptom treatments. And thirdly, to think about how we might predict early menopause, which could be useful for in lots of areas. So first of all, the new biology. So one of the challenges when we're doing a GWAS, so this is our Manhattan plot, you may remember from the GWAS, the 290 signals. When you zoom into one of those signals, what you see is something like this. So here again, each of these dots is one of those SNPs, but you'll see there's multiple SNPs all equally associated. And that's because there's a lot of correlation between those SNPs. But the problem is they lie over these genes. So here are all the genes in that region shown down here. There's lots of them nearby, and even over that peak, you've got, 5 or 6 different genes. So it's a real challenge, actually, going from these GWAS findings to understanding what are the genes that are involved. And we use a variety of techniques. We can go back to the laboratory and start testing things. We can use a lot of computer algorithms to predict. There's lots of ways that we can do this, but that's definitely an area that we need to progress further on. In this particular case, we had this gene underlying this peak, BRCA1, which you may know as the breast cancer susceptibility gene. And what we found was, when we started looking under all these peaks, we found a lot of genes that were involved in cancer, and particularly in particular processes that are important in cancer. And these are the processes of DNA damage repair. And so what we did was we looked at all of our 290 signals and we identified 3 key processes that were being affected by the different genes that were highlighted. We have repairing of double-stranded breaks. So when the DNA gets broken, there's a whole suite of proteins that are involved in repairing that. And the genes that were involved were actually active across the whole lifespan, from fetal life right up to adult life. There are a bunch of genes that were involved in the meiosis, the production of the gamete stage. So that's happening early on in development, before birth, predominantly. And then another large number that are involved in this atresia, the apoptosis process, so that decline from 7 million. So these processes are really key, and about 2/3 of the genes we identified were part of these different processes, these 3 processes. So the second area is therapeutics, and I wanted to use the example here of a study we've done looking at hot flushes or vasomotor symptoms, and this is a study that Kate's very much led, and we wanted to look at the genetics of hot flushes. As I said, about 70% of women have them, but were there any genetic predispositions? It's slightly harder to assay hot flushes in some of the studies we look at because it's not as well captured, as in, people aren't asked as often what their menopausal symptoms are. But in the UK Biobank study, which is a study of 500,000 people in the UK, they also collected GP records, so primary care health records, on about half of them. And what Kate did was to go through those records and pick out people that had these particular codes, so things like flush or flash in the GP record. And from this was being able to pull out about over 14,000 women who had reported to their GP that they'd had some sort of hot flush symptom. So now we've got this, we call it a phenotype. We can now get a bunch of controls as well that have been to their GP but didn't report these symptoms, and we can do one of those GWASs. And this is what we found. We only had one signal, but it was an exciting one. So this is the signal that we had, and it was right over the top of a gene called TACR3. It was a really strong signal, very strongly associated with hot flushes or vasomotor symptoms. And one of the reasons that this is so exciting is because TACR3 codes for a gene called NK3R, neurokinin 3 receptor. And this is a gene and a pathway that is a known drug target for hot flushes. And so this shows you where the NK3R protein interacts with the neurokinin B, its receptor, within the brain. And this is in the hypothalamus of the brain now. So we're not in the ovary anymore. We're looking in the brain, and these are neurons in the brain. And it's thought that the lack of estrogen that accompanies menopause causes hyperactivity of these neurons in the brain, such that more neurokinin B is produced, and that stimulates the neurokinin 3 receptor, which is involved in these neurons, which are important for thermoregulation, so heat regulation, and that's what causes the hot flushes. And this has been shown in studies where women have been given the neurokinin B, and that induces hot flushes. And they've also been given drugs that block this NK3R. They're called antagonists, and they reduce hot flushes. So our signal from the genetics, although this was known before we developed the signal, this is a really good proof of principle to us that our genetics can find these drug targets. And even more excitingly, one of those targets, there are about 3 or 4 of them, I think, that have been in clinical trials. And in May this year, the first of those has actually been approved to be used for treating hot flushes. So we're optimistic that by finding more of these signals, we might be able to find new treatments for hot flushes, which is really important because many women can't take hormone replacement therapy or don't want to. And so to have some alternatives would be really fantastic. The third element is prediction. And as I said at the beginning, the ovarian reserve is finite. So it's very much a storage cupboard, not a production line. So it's unlike sperm, where it is more of a production line. So knowing how big that store cupboard is would be really beneficial to women. At the moment, the methods to predict the size of the ovarian reserve are not particularly good. The best predictor, in fact, is knowing when your mother went through her menopause. Otherwise, you could use transvaginal ultrasound of the follicles, counting the follicles, not something you're going to do every day, or measure hormones. But actually, hormones only really change in their level when you get near menopause. And if you're looking at a fertility predictor, it may actually be too late by the time you can measure changes. So we've been looking at whether we can use genetics to be a better predictor. So in this diagram, you can see we've grouped together all those 290 genetic variants into something that we call a polygenic risk score. And we've looked at that polygenic risk score in women who had menopause before 45, that's in blue, and over 45 in orange. And you can see that there is a difference in the genetic risk score of those two groups. And this gives us hope that we can start to use these scores as a way of predicting whether women are more or less likely to develop early menopause. Obviously, we'd like the gap between these two profiles to be much wider, and hopefully by discovering more of the genetic component, we'll be able to do that. Another way of looking at this is to look at the risk of having very early menopause or menopause before the age of 40. This affects about 1% of the general population. But if you take women who have a high genetic risk score, the top 1%, at this end here, you're 5 times more likely to have menopause before 40 than women with the average genetic risk score. So this is start to become useful, actually, as a clinical tool, potentially. So we see 2 areas that this could be useful. Firstly, perhaps during infertility treatment, where it's really important to know how big the ovarian reserve is, to tailor treatment, to get the maximum number of eggs without overstimulating, which can be very dangerous. So if we can use genetics to inform that, that would be great. And secondly, to inform young women who are looking to plan their lives and their reproductive choices and may want to choose to freeze eggs or embryos if they know that they are at high risk of an early menopause and therefore early infertility. So just to sum up, I think hopefully I've convinced you that reproductive aging is governed by a mixture of genetic and non-genetic factors. the reproductive lifespan is predominantly driven by DNA damage response, although there's certainly other important pathways as well, and that we can use these genome-wide studies to uncover some novel biological mechanisms. Not only that, genetics can inform us about potential therapeutic targets like the recently approved menopausal symptom one, and also to start to predict which women are more likely to have a low ovarian reserve. And finally, just to acknowledge the people that are involved. As I said, all of these studies involve huge numbers of collaborators. So I've put them under the banner of the Reprogen Consortium here. So a lot of our papers have 300 plus authors, and we're working with studies across the world. So there's a lot of people involved. But our key collaborators are here in Exeter, at the University of Cambridge, led by John Perry, and at Copenhagen, led by Eva Hoffman. Thank you.
So Professor Murray's happy to answer some questions. So who would like to open the questions? Well, you're thinking about that. Let's go one
Okay.
Get things off. You mentioned that a lot of the genes were related to sort of fixing bits of DNA.
Yeah.
So do you think menopause is, or earlier menopause is just, there's a general aging going on the menopause is just a marker of that general aging, or is it a completely separate process?
It's a massively hot topic, Craig. So there's a lot of people looking at that. So the aging field and the reproductive aging field are very much looking at the same things. So there are definitely overlaps with the two. It's very hard to say. I mean, so one of the markers might be mortality. So you might predict if it is about general body aging, that women with earlier menopause might die earlier. And there is some evidence for that, but I don't think it's that convincing, and doing those sorts of studies is difficult. So I don't think we know for certain, but I think there's probably some overlap and some differences is what I would predict. Yeah. Oh, lots of
Have we got a question there first? Then you were first.
You mentioned that there's quite a lot of species that don't go through the menopause.
Yeah.
So are you doing comparisons with those? Are you looking at the genes you identify and seeing if they're different in those species to try and understand which ones might be important or more important?
We'd love to do that. The problem we have with other species is getting the variation. So to find these variants, as I said, we needed 200,000 women. It's very unlikely you'll get 200,000 killer whales, for instance, or 200,000 dolphins. So that's one of our challenges. But we do have ideas to look at those similar, what we call homologous genes within those species, and to see whether some of the changes that are... So one of the ideas was to compare the dolphin and the killer whale, for instance, for the genes that we are identifying. So is there evidence that the killer whale has evolved the same genes? It's a bit of a long shot, though, because the process might be very different in a killer whale. We suspect that it's almost certainly that if it is a positive evolution, that it's happened independently in the two species. So I'm not sure, but it's definitely in the mind, yes.
Who's next? Your hands up. Got one down here?
Hi, thank you. You mentioned the test for ovary reserve to predict fertility or fecundity, and so to what extent understanding the gene role would improve? I mean, is the ovary reserve not a very accurate test for timing of menopause or for fecundity?
Do you mean like in current IVF practice? Is that what do you mean?
Yes, in this context, yeah.
Yeah. So I think what's used currently is hormonal measures and ultrasound, but there's definitely cases of ovarian hyperstimulation, for instance. So women are given too much of the stimulating hormones, and so I don't think this would replace those. I think it could be a part of that test, and we are actually testing that at the moment, whether it could refine the existing measures, because certainly hormones are important, but we know that hormones you might get quite different results on different days, for instance, and there's sort of variability in there. So it could be part of the suite of things that get measured as part of IVF. So that's something we're looking into, yeah.
Okay, thank you.
Fabulous talk, Anna. I'm interested in you saying that timing of puberty is one of the factors which actually sets timing of menopause. I mean, is having puberty early or late, like there's a stopwatch that goes on? I mean, so what do we know actually about when puberty occurs and how that actually influences menopause?
Yeah, I don't think it's a very strong predictor, actually. And certainly in our first studies that we did, there wasn't much overlap between puberty and menopause. But I think we're more and more convinced that there is a relationship, but it's not strong. And certainly the genetics of puberty is very different from the genetics of menopause. So the puberty timing genetic studies, which we've been involved with as well, but as you know, led a lot by Ken Ong in Cambridge and John Perry, that they're showing that a lot more involvement of brain-derived factors and obesity is a very key driver of puberty timing. But there are certainly overlapping genes, and they tend to be the hormonal type genes. So things like the follicle-stimulating hormone gene is involved in both traits. So I think that could be where the overlap comes from, some of those overlapping pathways that are involved in the two processes. I mean, it may be confounded by obesity. I'm still not convinced of the role of obesity actually in menopause. I mean, there's studies going both ways. So we're not completely convinced by those studies at the moment, but I wouldn't be at all surprised if obesity was involved.
Yes. Here and then to
Hi, I was wondering, because you're doing whole genome sequencing and you've seen a lot of pathways linked to double-strand breaks, are you going to be looking at structural variants and perhaps the susceptibility to that, which might add to the genetic variation you're seeing?
Yeah, absolutely. So that's definitely a class of genetic variant that we haven't captured very well so far, particularly with the GWAS, as you probably know, it's not particularly well captured. But yes, by doing the whole genome sequencing, we'll be able to look at that. And, things like, so when I first started working in this field, I worked on the Fragile X syndrome, which is a kind of a structural variant. It's a trinucleotide repeating motif, so it gives you a large repetitive region in the genome. That was one of the first genes that was associated with earlier menopause. So it would be really great to assay that at scale, that particular one, and there may be other examples of similar types of structural variant that will have a big effect. Yeah, definitely. And I think probably things like, we know that things like Turner syndrome, which is loss of a whole or part of the X chromosome, which I guess you could count as a structural variant, especially deletion, is associated with earlier menopause as well. So.
Thank you for a great talk. I have two questions, if that's okay.
Yeah.
First one is, I know that there's been significant differences found between different ethnic groups for puberty onset. I was wondering if the same has been observed for menopause. And then the second question was, is there any effect of the combined pill or other birth control on menopause onset?
Oh, great questions. Thank you. I think there's some evidence of differences between ethnic groups for menopause, but it's nowhere near as strong as it is for puberty. And I think one of the main differences is, as I mentioned, to answer the previous question, that a lot of the puberty determinants are around body size and body shape. And fat content of girls and the size. So there are lots of ethnic differences in body shape and size that I think would account for those differences in different ethnic groups. As for the role of the contraceptive pill, it doesn't seem to have any influence on timing of menopause. And I think that's because the contraceptive pills are just preventing ovulation. And as I said, ovulation actually is a drop in the ocean when it comes to the number of eggs that are lost in the ovary. So actually, if you stop ovulation, it's not going to have a dramatic effect on the timing of menopause, and that seems to be the case from the data.
Good. Question here. Is that question there?
Sorry, me again.
That's okay.
Has anyone looked at mitochondrial genes?
Mitochondrial genes?
Yeah, and how that might alter loss, atresia, or anything like this and affect menopause?
As far as I know, no, I don't think there's been a big study of mitochondrial genes, but again, we will capture that with the whole genome sequencing. So again, that will be something that we will be looking at. We have looked at the first, so the UK Biobank have released whole genomes on 200,000 people. So we've had a quick look at that. The problem with whole genomes is it's much harder to find things because you've got so much more to look at. So a lot more things will come up by chance. So working out the real from the not real is much harder, but we'll certainly will be looking at the mitochondria. We had a quick look at the mitochondria and found nothing so far, but I'm not aware of anything published about mitochondrial associations, no.
Lovely. Well, in the interest of time, and just before we thank Professor Moran again, just let you know that we are having a wine reception outside, and you are all welcome to stay and enjoy a glass of wine.